we're diving into the fascinating world of phospholipids, the key molecules that make up the fabric of cell membranes. So, understanding their structure is essential because it explains how our cells protect themselves, interact with their environment, and maintain internal balance. So, by the end of this lesson, you should be able to first identify the basic structural components of a phospholipid. Second, describe the amphipathic nature of phospholipids. Third, differentiate the types of head groups based on their chemical structures. And fourth, interpret the general structural formula of phospholipid as shown in chemical diagrams. And keep these outcomes in mind since we'll revisit them later at the end of our discussion. Now let's start with the big picture. What are phospholipids? So phospholipids are crucial molecules in biological systems. Their main job, they form the structural foundation of cell membranes, which means they create the boundary between the cell and its environment. And thanks to their unique chemical structure, they form barriers and control what enters and exits the cell. So please refer to the diagram shown. So the diagram shows a simple model of a phospholipid. So as the small circle on top represents the polar head, while the two long tails represents the non-polar fatty acid chains. And it's this combination of parts that gives the phospholipid its unique properties. With that being said, let's break that phospholipid down into its basic parts. So a phospholipid is made out of first, a glycerol backbone, two fatty acid tails, a phosphate group, and a polar head group. So what are these? So glycerol backbone is a 3-carbon molecule that acts as the spine that holds everything together. The two fatty acid tails, these are long hydrocarbon chains that attach to the first 2 carbons of the glycerol. Third, the phosphate group, is the one that attached to the 3rd carbon of the glycerol, and this part carries a negative charge. And lastly, the polar head group. This connects to the phosphate and can vary, such as choline, serine, or ethanolamine. So as you can see in the image shown, the head is represented by a round shaped or ball. So this part is hydrophilic. The 2 tails naman are wavy lines that are the fatty acid chains and are hydrophobic. And these 4 components are what gives phospholipid their structure and function. Now let's look more closely at the structure. So the head of the phospholipid is made out of glycerol and the phosphate group. And together, these make the hydrophilic, also known as water-loving, or the polar part of the phospholipid structure. And then the 2 tails are 2 fatty acids that are non-polar and hydrophobic or water-fearing. And then these 2 fatty acid chain, one is usually saturated or straight and the other is unsaturated, which has a kink due to a double bond. So the polar and non-polar parts, this difference is very important since it means that phospholipids are amphipathic. And that's a word we'll keep repeating all throughout this lecture video. The term amphipathic is key to understanding phospholipids. It means the molecule has both polar and non-polar parts, like what I've said earlier. The head is polar and interacts well with water, and the tails are non-polar and repels water. But why is this important? So in water, phospholipids automatically arrange themselves into a bilayer, with heads facing the water and tails facing inward, or away from the water. And this forms the cell membrane, which acts like a skin for the cell. Now, as you can see in the picture shown, the picture shows a bilayer, where 2 rows of phospholipids face opposite directions. This is how your cell membranes naturally organize. With that being said, we now reached our first checkpoint. So I would like you to discuss it with your classmate, or if you're feeling shy, you can write your answers on a piece of paper so you have something to review. So checkpoints are placed all throughout the lecture video for you guys, for all of you to be engaged in this video or in this lecture. So for our checkpoint 1, we have 2 questions. So first question, which part is hydrophilic and which is hydrophobic? And the second question, what would happen if both ends were hydrophobic? I will give you 5 seconds for that. Now, let's talk about the glycerol backbone. So glycerol is a 3-carbon molecule, and each carbon bonds to a different group. So carbon 1 and carbon 2 bonds with the fatty acid chains. So carbon 1 bonds with the saturated fatty acid and carbon 2 bonds with the unsaturated fatty acid. And then carbon 3 bonds with the phosphate group. So look at the diagram. So this structure forms the core of the phospholipid. Now for our second checkpoint. So how many carbons are in glycerol? Second, what are the 3 components attached to each carbon of the glycerol backbone? So after that, next are the fatty acid tails. So these are long hydrocarbon chains. So as you can see in the image, notice how one tail is straight and the other is bent, right? That's because of the type of bond in the chain, as what I've said earlier. So one is saturated, which means it's a straight chain with no double bonds, and one is unsaturated, which is a bent chain and has a double bond. And then these differences affects membrane fluidity. Membrane fluidity is how flexible or stiff the membrane is. So the saturated fatty acids are packed lightly, so it has less fluid membrane, while the unsaturated fatty acid has an increased membrane fluidity since the kinks in the chain prevents tight packing. With that being said, we've reached our third checkpoint. So first question for our third checkpoint, what is the difference between saturated and unsaturated fatty acids? And then the second, how do fatty acids affect membrane fluidity? Now let's move on to our third component. So the phosphate group. Let's now focus on the phosphate group. So this group is negatively charged and polar. So it's negatively charged because it loses hydrogen ions, if you may ask. So the phosphate group attaches to the 3rd carbon of glycerol and makes the whole head region hydrophilic. So you can see in the image, you'll usually see a letter P or a round symbol near the head. So the letter P represents the phosphate group. With that, we've now reached our 4th checkpoint. So first, which hydrocarbon or glycerol does the phosphate group attach to? And second, what charge does the phosphate group carry? And lastly, the head group. The head group is the final component. So it is attached to the phosphate group. And different head groups include choline, serine, and ethanolamine. So as you can see in this slide, so there are different substituents for the head group. For example, for choline, we have N positive. For ethanolamine, we have NH2. And then for serine, we have NH2 and COO negative. So that's where they differ. But what differences do they have compared to the others aside from their substituents? So choline has a neutral charge and is important for membrane structure and signaling, while phosphatidylserine or serine has a negative charge and plays a role in cell signaling and apoptosis. And lastly, ethanolamine or phosphatidylethanolaminehas a neutral to slightly negative charge and is important for membrane curvature and vesicle formation. So the head group determines the type of the phospholipid and how it behaves in different parts of the body. But note this, each head group can look a bit different chemically, but all are polar and water-loving. With that being said, we've reached our last checkpoint. So for our last checkpoint, what head group is attached to the phosphorus group? So the phosphorus group is here, and then the attached head group has an N positive. So what do you think is the head group for that? Now let's revisit the amphipathic nature of phospholipids and summarize everything we've learned. So hydrophilic head plus hydrophobic tails makes it amphipathic. So this unique structure drives the formation of the phospholipid bilayer, where the tails hide from water and the heads face the watery environment. So let's look at the bilayer diagram shown. Look at how the molecules are arranged. So the heads face the outside and the insides of the cell, while the tails form the inner barriers. And then this forms the basic structure of cell membranes, that's where they function. Now let's summarize everything we've learned. So phospholipids are made out of glycerol, 2 fatty acids, phosphate, and then a head group. And the second, they are amphipathic, meaning they both have water-loving and water-fearing parts, or polar and non-polar parts. And this is what allows them to form membranes and create boundaries in cells. And then lastly, the head group determines the type and specific role of the phospholipid. With those being said, let's check back to the learning outcomes. So first learning outcome, identify the basic structural component of a phospholipid. So can you now name the structural components? The 4 structural components of phospholipid. So we have the glycerol, the 2 fatty acids, the phosphate group, and then the head group. And for their second learning outcome or learning point, describe the amphipathic nature of phospholipids. So amphipathic means it both has water-loving and water-fearing parts, which is crucial for their function in forming cell membranes. And for the third, differentiate the types of head groups based on their chemical structure. So, the choline, the serine, and then the ethanolamine, right? So, choline, we have the N positive, and then the serine, we have the NH2 and COO negative, and then for ethanolamine, we have NH2. And then lastly, can you now interpret the general structural formula of a phospholipid as shown in chemical diagrams? Okay, great job everyone. Now, here are the references used for the lecture, and feel free to take a picture of it and do more research to gain further understanding. And please also do scan the QR code flashed on the screen, as this will take you to answer a 5-item quiz, which also serves as your attendance for the day. So, that concludes our lecture. Thank you for listening everyone, and until our next lecture together. Bye!